We still have some folks entering the room, so we'll give it another minute. Okay, we'll go ahead and get started. Good afternoon. We're very pleased to welcome all of you here today under some very different circumstances than from previous years. I'm Holly Anderson and I'm the Executive Director of the Breast Cancer Coalition. Most of you know our history, that we are a grassroots community-based organization and we're dedicated to eradicating breast cancer, a big initiative through action and advocacy. We formally organized as a 501c3 nonprofit organization in December of 1997, and we are the only independently operated full scope organization focused specifically on breast cancer in the greater Rochester region. 5 years ago, we began welcoming women with gynecologic cancers. We learned that there were many similarities between the circumstances and experiences in breast and gynecologic cancer survivors. And many gynecologic cancer survivors had approached us in the past, hoping to learn of a similar organization for them. We now serve women and a handful of men throughout the central and western New York regions of New York State. And as challenging as COVID-19 has made things, we have been able to continue and even expand these services thanks to people, groups, corporations like you, people who provide financial support for what we do. Breast cancer continues to be one of the most common cancers among women in New York State, and this is what initially compelled us to initiate a research program that focused on breast cancer. Each year in New York State, over 16,200 women are diagnosed with breast cancer, and about 2,500 of them die from the disease. It's estimated that 1 in 8 women will develop breast cancer in their lifetime. And additionally, about 150 men are diagnosed with breast cancer each year in New York State as well. Today represents a coming together of multiple institutions to assist us with our overarching goal, which is to end breast cancer through research that focuses on prevention or cure. And by cure, we mean that which halts progression of disease in a metastatic setting, and in best cases achieves regression and a status that we all long for, no evidence of disease, NED. Simply put, we don't want people to get breast cancer, and if they do, we don't want them to die of it. Most of you know that the Breast Cancer Coalition has a 4-pronged approach to the rising number of breast cancer incidences in our region. It's advocacy, education, support, and research. There's a lot of fundraising on behalf of the ubiquitous term breast cancer, and we see it everywhere, particularly during the Breast Cancer Awareness Month of October. But it's become more and more difficult to understand what happens to these funds, where they go, and who they go to. So when it comes to fundraising for research, we believe funds raised in our region should stay right here in our region. And we believe that the voice of the breast cancer survivor and other advocates should be heard when making decisions about where and how those funds are spent. Our breast cancer research initiative has represented a unique collaboration that includes breast cancer survivors, scientists, clinicians who hail from Cornell University, Roswell Park Cancer Institute, University of Rochester, and Upstate Medical in Syracuse, as well as survivor advocates from all of those areas. In each February, we convene a review panel that represents a balance of representatives from each institution, as well as survivors from that wider community as well. And while we appreciate the efforts of each one of our panel members, we're especially grateful to the breast cancer survivors who serve on that panel, often overwhelmed by the sheer science of it, but always reminding the scientists in the room why they're doing what they're doing and putting a face on breast cancer survivorship. It's our belief that breast cancer advocates who are trained, educated, and represent this particular patient constituency must be meaningfully involved in all aspects of decision-making that affect breast cancer research. And as of today, after this ceremony, we will have awarded over $975,000 to 24 researchers, including the 2 that you're going to hear about shortly. And before I turn this over to our research administrator, Helene Smither, I'd like to thank all of those who have contributed to this fund, to this initiative, either directly or indirectly, and especially the time that those have put into it. Thank you for supporting our programs that allow us to provide education and support programs to the thousands of breast and gynecologic survivors living in our region. And now I want to introduce Helene Smither, who is the absolutely amazing research administrator here at the Coalition. Helene herself is a breast cancer survivor, and she stepped forward 3.5 years ago to lead this effort. She has really shaped it in so many different ways. And it has been an overwhelming process to transition this whole project to an online virtual experience, from how we did grant review this year to this ceremony today. And so I would like to introduce Helene Smither. Helene.
Okay, thank you, Holly. Okay, the Coalition started its grant program in 2003. In 2017, the Coalition awarded its first grant for pre- or postdoctoral students to support outstanding graduate students or recent PhDs launching their careers. Including today's awards, the Coalition has awarded 24 grants for a total of $976,125. Each fall, the Coalition sends out a request for proposals to regional academic, medical, and research institutions. This year's applications were assigned to a review panel of 10 scientific researchers and faculty members and 8 breast cancer survivors who are members of the Coalition's research committee. The panel met virtually on Zoom for a full day in February to jointly discuss and review the proposals and recommend which proposals should be awarded Coalition funding. I'd like to thank the review day participants for their work. Dr. Scott Gerber, Dr. Helene McMurray, and Dr. P.J. Simpson-Hyderis from the University of Rochester, Dr. Kelly Madden from the Brockport Research Institute, Dr. Jihong Gong and Dr. Chi-Chen Hong from Roswell Park Comprehensive Cancer Center, Dr. Robert Weiss, Dr. Kelly Hume, and Dr. Mark Antoniette from Cornell University, Dr. M. Saeed Sheikh from Upstate Medical University, and research committee members Wendy Bachhuber, Dr. Gunhilda Buchsbaum, Beverly Cannon, Allison Curry, Kathy Finnegan, Nancy Granke-Cure, Wendy Godorf, Julie Overbeck, Kathy Simpson, and Jean Verhulst. Their recommendations were presented to the Coalition's Board of Directors, who approved the awards. And now I'd like to ask Holly to announce the names of the grant recipients.
I had to unmute myself, technical little thing there. Let's see here. So I'm pleased to announce that this year the Coalition is supporting 2 young scientists who are starting their careers. Each will receive a 2021 grant for pre- and postdoctoral trainees in the amount of $25,000. The review panel felt that their proposals were innovative and would have high impact in the field of breast cancer research. The first grant is being awarded to Dr. Sagar R. Shah for his proposal, Identification and Functional Characterization of Enhancer-Associated Mutations in Breast Cancer. Dr. Shah is a postdoctoral associate in the Department of Molecular Biology and Genetics at Cornell University. The second grant is being awarded to Tao Dai for his proposal, Uncovering the Implications of Breast Cancer Metabolic Adaptation in the Tumor Microenvironment. Tao is a 3rd-year PhD student and a pre-doctoral trainee in the Department of Immunology at Roswell Park Cancer Institute. We congratulate Dr. Sagar R. Shah and Tao Dai and are proud to present them with the Coalition's 2021 grants to support their work.
Thank you.
Thank you very much.
Helene?
Okay, I'll introduce the recipients who will explain their research proposals. Our first recipient is Dr. Sagar R. Shah. Dr. Shah is an ethnic Indian who immigrated from Tanzania, Africa, to the United States with his family when he was 15 years old. He entered a 5-year program at Clemson University, where he received his Bachelor of Science with high honors in biological sciences and his Master of Science in bioengineering, earning the school's most prestigious award, the Norris Medal. Dr. Shah was selected to be the student speaker for the doctoral commencement ceremony when he received his PhD in biomedical engineering at the Johns Hopkins University School of Medicine. He then pursued a joint postdoctoral fellowship at Johns Hopkins and the Mayo Clinic. Dr. Shah is the recipient of numerous other honors and awards, including a National Science Foundation Graduate Research Fellowship. He has co-authored 22 peer-reviewed manuscripts, 10 as the first or co-first author. Dr. Shah has also co-authored 4 patents on the design and the development of therapeutic agents, targeted drug delivery systems, and prognostic devices for the management and treatment of cancer. Aside from his research in the lab, Dr. Shah serves on the scientific advisory board of a biotech startup focused on combating aggressive solid tumors. Currently, Dr. Shah is a joint postdoctoral associate in the laboratories of Dr. John Liss and Dr. Hei-Yuan Yu at Cornell. If you have any questions for Dr. Shah, please submit them through the chat feature, which you'll find at the bottom of your screen. And I will read them to Dr. Shah when he finishes his presentation. So, Dr. Shah, you will now have control of the screen. Thank you.
Hi. Can you see my screen before I get started?
Yes, we can.
Great. Well, thank you very much. Thank you for the kind introduction. It's an honor, a deep honor for me to be here today. And I'm very grateful for the coalition's support of my proposed work. Before we get started, please refer to me as Sagar. I usually do not respond when I hear Dr. Shah. I usually think it's for my dad. So I'm not that old yet. Anyway. So as many of you know, identifying critical mutations that drive breast cancer development and progression has been a persistent challenge in the field. Today, I'll give a quick overview of our current understanding, some of our outstanding questions in the field, and my approach of addressing these questions through my research. Now, DNA sequencing of patients diagnosed with breast cancer and the tumors themselves have revealed a plethora of mutations. These mutations and changes in the underlying DNA code can either be inherited from our patients and thereby can increase an individual's susceptibility to cancer, or as seen here in the second figure, they can occur for the first time in the cells themselves and lead to tumor development. Now, in general, these cancer cells can have more mutations than normal cells, but each person's cancer has unique combination of these alterations. Now, interestingly, a bulk of these mutations reside in a largely obscured part of the genome called the non-coding genome. Now, what is the non-coding genome? Well, to understand that, let's take a step back and look at the coding genome first. As many of you may be aware, our body is composed of trillions of cells, Within each cell, you'll find the blueprint for constructing and operating the human body, stored in sequences of DNA called genes. This genetic information is used to make proteins, which are the basic, yes, basic fundamental blocks of cells that dictate cell function. However, these protein coding genes only account for 2% of the genome. Yes, that's right. That leaves behind, sorry about that, that leaves behind 98% of the genome. Yes, that's the majority of the genome. This uncharted DNA region, long thought to be useless, was commonly referred to as junk DNA. However, new research is revealing that these junky parts of the genome, in fact, play important roles, but are just poorly understood. So indeed, accumulating evidence indicates that sprinkled within this junk DNA are important sequences called enhancers, as labeled here in this figure. These enhancers are integral controllers of genes and thereby cell function and behavior. Now, it's estimated that there are hundreds of thousands of enhancers in the human genome. Yes, that's right. There are more enhancers in the genome than protein coding genes. Thus, it's not only important to identify and define these critical mutations or elements, but understand their function in cancer. Given their critical role, mutations in these enhancers could dramatically alter cell function and cell behavior. As seen here, a critical mutation in any of these enhancer elements can turn off a gene such as a tumor suppressor and therefore allow the cells to grow very rapidly. Alternatively, these mutations in the enhancer elements can turn on a cancer-driving gene, thereby promoting the growth of the cancer. However, in many cases, enhancer mutations, as seen here, may not have any observable effect or any alteration in gene function. Thus, it's important to systematically identify and characterize enhancer malfunction in breast cancer. In our opinion, a rigorous analysis will allow us to narrow down our search and identify those driver mutations that are critical for breast cancer progression and pathogenesis. However, this presents a needle in a haystack problem. How so? Well, because these mutations are peppered throughout the vast non-coding genome, and because enhancers represent only a subset of this non-coding genome, and moreover, because not all enhancer mutations impact cell function and disease state the same way, thus, because mutations that affect breast cancer are needles in a haystack of mutations in this junk DNA. Therefore, a new approach is warranted. Fortunately, I have access to some of the state-of-the-art technologies and methodologies that have been developed in my labs to precisely tackle this problem. For example, we have methods that can truly drill down into the genome and identify these active DNA elements, including enhancers, at high resolution and specificity. In addition, we have large screening methods to quantify and test enhancer activity en masse. That's right, that's hundreds or thousands at a time. This means that we can take these enhancer sequences and interrogate them in several ways, such as the effect of mutations on their activity, regulation of cancer-driving genes, or response to therapy. Now, using these tools, I'll comprehensively map the active enhancer landscape of breast cancer. Given my use of a large panel of breast cancer cell lines and patient-derived tissues, I will also seek to identify any subtype-specific enhancer profiles. This may be useful for clinicians in the future as a biomarker tool when detecting early onset breast cancer or metastatic potential. Altogether, these approaches will allow me to precisely pinpoint and characterize critical mutations in these active regions, both from the inherited or acquired types, with a particular focus on triple-negative breast cancer. Now, ultimately, this research will not only provide insights into the cause and drivers of breast cancer, but guide the development of highly specific drug therapies to improve patient outcome. And therefore, funding from this coalition will get us a step closer to finding that needle in the haystack. Thank you very much for this opportunity to present my proposed work. And more importantly, I can't thank you enough for your generosity, which will allow us to not only in our pursuit of answering some of these complex questions, but may allow us to maybe get a step closer to identifying some of the true culprits, meaning the mutations in these desert of our genome that will have a critical impact on the progression of this cancer. And it will allow us to inform new novel therapies that eventually we hope will be highly specific and can help us curb this aggressive cancer, including triple negative breast cancer. With that, I would like to acknowledge my labs, both John Liss and Haiwan Yu, who are my co-mentors, and individuals in our groups who have played an important role and will guide me through the process as we move on. And my collaborators, especially Charles Danko and Andrew Clark at Cornell. With that, I'd be happy to take any questions that you may have, and I hope I've done a decent enough job to convince you of what the deficits are in the field and how our approach will be able to tackle those problems. And I look forward to learning more about the cancer and finding ways to interrogate them, but also trying to find ways to stop and curb, as Holly mentioned earlier, trying to put the halts or the brakes on this cancer so we can get to a curative model. Thank you very much.
All right, thank you, Sagar. I have a communications background, and I always come, I have always done whenever I've interviewed people, I've come to them asking, how did you, one of my first questions would always be, how did you come to do this kind of study? What was it that drove you into this particular issue?
Great. Yeah, sure. Let me share some of my experiences. So apart from my training that has at least helped in feeding my intellectual curiosity about cancer development, I have a deep-seated interest in this because of some personal connection to the disease. When we first immigrated to this country, one of my aunts, my mom's sister, who resided in the U.S. at that time, was diagnosed with breast cancer, and along the same period, my paternal aunt was also diagnosed with breast cancer, and both of them had very different progressions in that cancer, and eventually both of them died from fighting the cancer. And so I have an interest as a personal connection to it, but I'm also very fortunate to have the opportunity to be able to try to understand this disease at a more fundamental level. And maybe through some of my work, I hope that I can take it to a level where it can be useful and beneficial for the scientific and the medical community. Thank you.
Thank you. Did you start your research when you came to Cornell, or had you started it at Johns Hopkins?
Yeah, so while I was at Hopkins, most of my work was focused on brain cancer, including metastatic breast cancer that oftentimes will migrate to the brain. And during that time, I think it was during that transition and coming to Cornell was when I truly invested all of my efforts into understanding breast cancer.
Okay, all right. Thank you, Sagar. I think if there aren't any other questions, I will go on, and I will, oh, wait, there is a question from Holly Anderson. If this Sagar, if this project yields the expected results, where will this lead?
Yeah, that's a great question. So our first steps are not just mapping the landscape, but then trying to figure out whether any alterations in this will have any effect on gene function. Ultimately, what we would like to get to is the third phase that I see is where we can start manipulating some of these sequences and testing it in cells and eventually in a mouse model to see whether they truly have a consequence on the physiology of the cancer. One thing that I mentioned towards the latter part of my presentation is that I hope by profiling these cancers, we can start maybe identifying early biomarkers too. So there are a couple of ways that I can envision this to go and progress as we learn more about the enhancer landscape and the mutation profile of it. I hope that answers your question.
Okay. All right. Okay, thank you, Sagar.
Thank you very much.
Okay. I will now introduce our second grant recipient. Our second grant recipient is Tao Dai. Tao Dai received his Bachelor of Science degree in Pharmaceutical Sciences at the School of Pharmacy at Fudan University in Shanghai, China, where he completed his undergraduate thesis in the lab of Dr. Yi Zhen Zhu. At Fudan, Tao was awarded several scholarships for his academic achievements and worked as an undergraduate research assistant. He also worked as an assistant analyst in the antibiotics department of the Shanghai Institute for Food and Drug Control. Tao then joined the laboratory of Dr. Subhamoy Dasgupta at Roswell Park Comprehensive Cancer Center in Buffalo to pursue his PhD degree in cancer sciences. Tao has already published 5 co-authored papers, 3 papers as an undergraduate student, 1 as a rotating graduate study, and 1 in his current lab. Again, if you have any questions for Tao about his presentation, please enter them in the chat feature, and I will ask Tao at the end of his presentation. Tao, you have control of the screen now.
Can anyone see my the presentation? Yes. Okay. So hello everyone. My name is Tao, and I'm a third-year PhD student in Dasgupta Lab at Roswell Park Comprehensive Cancer Center. So first of all, I'd like to thank Helene for the very kind introduction, and also the Breast Cancer Coalition of Rochester for the research support and for the giving me the opportunity to present my work today. So the title of my talk is uncovering the implications of breast cancer metabolic adaptations in the tumor microenvironment. So I like to start with a bit of background about breast cancer. So as some of you may know, that the female breast cancer has become the most diagnosed cancer around the world in 2020, based on a very recent report published this year. And another report published this year has suggests that the breast cancer will remain the top 1 diagnosed cancer for females in the United States in 2021. And although we know that the overall 5-year survival for breast cancer is around 90%, we are still facing some very critical challenges in the clinic. And one of them is the breast cancer spreading, which is also called the metastasis. So most breast cancers start from the duct that carry the milk in the breast. And so initially the tumor cells grow within the lining of the duct. And this is called the ductal carcinoma in situ. And over time, some tumor cells become more aggressive, and they can manage to break the wall of the duct and to spread to the surrounding breast tissue. So this is called the invasive ductal carcinoma. And then some tumor cells can find their way to enter the circulation, including the bloodstream and lymph, which is called the circulating tumor cells. And those circulating tumor cells that can survive in the circulation will eventually spread to the distant organs such as bones or lungs, which is hard to treat in the clinic. So my project aims to answer the questions: how do those tumor cells spread from the primary site, and how to stop the spreading of those breast cancer cells? So now let's zoom into the primary tumor microenvironment. So here we can appreciate that the primary tumor is not just made of just tumor cells. It's actually a mix of different cell types, including the cancer cells, immune cells, stromal cells, and also the blood vessels. And as the tumor starts growing, all the cells need to compete for very limited nutrients and oxygen. So even the tumor cells need to compete with each other for very limited those nutrients. And so even for the tumor cells themselves, it's a very harsh environment to live in. But some tumor cells, they can manage to get adapted to this harsh environment. And some tumor cells can even change the microenvironment by secreting the tumor-derived factors to support their own growth. And we believe that these tumor cells that can adapt to the harsh primary tumor microenvironment will have a higher potential to survive and grow as they spread to the circulation or distant organs. And our lab focused on one of the ways that tumor cells can use to adapt to the environment, which is through a metabolic enzyme, PFKFB4. So this is an enzyme that highly expressed in breast tumor cells, and it can help the tumor cells to sense the glucose level and oxygen level in the tumor microenvironment and use the glucose as the source of energy to help the cells to survive and grow. And our lab found that this enzyme can do more. So we have some preliminary findings to support the hypothesis that the PFKB4 can help the tumor cells to escape this harsh tumor microenvironment and to migrate towards the blood vessel where there are sufficient nutrients and oxygen supply. And as they move closer to the blood vessel, they're more likely to enter the circulation and spread to distant organs. And on the other hand, we also found that the tumor cells with high PFKFB4 expression can somehow suppress the cytotoxic T lymphocytes, which is the major immune cell type that can attack the tumor cells. So these are the two arms of my proposal that I'm going to test in the near future. So eventually, the ultimate goal is to test whether inhibiting PFKB4, either alone or in combination with other treatments, for example, immunotherapy, can reduce the tumor cell migration to the blood vessel and to restore the immune cell function, which ultimately stop the breast cancer spreading. Yes, so in the end, I would like to thank my mentor, Dr. Dasgupta, and my co-mentor, Dr. Abrams, for their comments and suggestions, which are incredibly important for developing my project. And I would also like to thank all the members in the Dasgupta lab, as well as the collaborators and share resources at Roswell Park, and also the founding that support my preliminary work. And with that, I'll be happy to take any questions. Thank you.
Okay, Tao, I'm going to ask you the same question that I asked Sagar, which is, how did you come to this particular line of research and study? Is this something that started in your undergraduate work or has developed over the past 3 years of your graduate studies?
So I have actually have two experiences in my undergraduate study focused on the breast cancer. So the first one is more on how the mTOR signaling affect the lipid metabolism in triple negative breast cancer. And another one is how the other one is when I was doing the rotation at Roswell Park, and I did that rotation in the clinical lab, which is under the supervision of our the chief of breast surgery, Dr. Takabe's lab. And then from there, I learned like how to think from a clinical perspective, how to like initiate the study from the clinical database. And from there, I learned what's the major like challenges that the clinicians are facing in the clinic, which is the metastasis. And then, yeah, I started my project in Dr. Dasgupta lab, and he's a wonderful mentor. He has a really great experience in breast cancer biology, and I hope I can develop my project here. Yes.
Okay. All right, thank you. We have a question. Tao, what is the structure in your diagram labeled CAFs?
Oh, CAF is cancer-associated fibroblasts, which is so in the tumor microenvironment, there are different subtypes of different types of cells like immune cells and fibroblasts. And fibroblasts is one of them. And when we call it cancer-associated fibroblasts, that means those fibroblasts receive some kind of signals from the cancer cells and start to get reprogrammed and help the tumor cells to survive and live inside the tumor. Yes.
Okay. All right. Well, right now it looks like there are no more questions. So, oh, wait, wait, of course they're coming in right now. One question for both, to both of you is, well, let me ask you a second question first, because I believe this is towards you, Tao. If cancer cells enter the bloodstream to obtain glucose and nutrients, why do they leave the bloodstream?
Okay, so in the bloodstream, we can consider bloodstream as a completely different microenvironment. So there is enough nutrients and oxygen, but the tumor cells will face other stress, such as there's a physical, think about the fluid, the blood is pumping, and then there is like a physical stress on the tumor cells. And also there are studies showing that inside the blood, the tumor cells are facing more the oxidative stress, which is also that is something that the tumor cells need to overcome in order to survive. So, and also they're saying that in the bloodstream, the tumor cells are not circulating as a single cell. So some tumor cells will like, they form a cluster together with other cells in order to survive. So which means that the tumor cell as a single cell, it's not that easy to survive in the bloodstream. Yes.
Okay, all right, thank you. And this is a question for both researchers. As researchers at the early part of your careers, where do you hope to be in 5 years? Sagar, I'll ask you that question first. Where do you hope to be in 5 years?
Oh, well, that's a question, I guess, sometimes when you're in training, you always try to avoid at some phase in your training. I'm hoping that at some level, I think I would be well suited to start thinking about my next step, which would be either in an academic institution, if an opportunity presents, or in a biotech environment where I can take some of the ideas that we find on our bench side and try to find ways to translate that and bring them to the bedside. And so both of those options are still, to me, attractive. How things will develop in 5 years, it's still unclear. Ever since I was a kid, I've always had an evolving perception of what my life would be in the next 5 years. So I'm open-minded, but at the moment, I'm focused on the work and committed to answering some of these questions.
Thank you, Sagar. And Tao, where do you hope to be in 5 more years?
I think I have a very solid plan of my next 5 years. So yeah, I like Boston a lot. So I'm already started to hunt for some very good postdoc position that kind of match with my interest in breast cancer field. And then hopefully after I graduate from here, I can start my new career as a postdoc in Boston. And then hopefully 5 years, yes. Yeah, eventually I would like to start run my independent lab. Yeah, that's my dream. Yes, hopefully that can come true some. Yes.
Okay, thank you. Well, we have no doubt that each of you will be very successful, and we will be hearing more about you over the next 5 years and after 5 years. And we wish you both much future success and much continued success. And so right now, I just want to thank everyone who's here today for attending the grant announcement today. And I'm going to turn this back over to Holly for some closing remarks.
I muted
Holly?
myself. I gotcha. As I was sitting here listening to them, I realized that I had back here on the back of my desk, these are this year's grant proposals. So I just wanted to share the depth of the submissions this year was extraordinary. And listening to your talks, your presentations really gives me, as a survivor of stage 3 breast cancer, hope. And I know that others here today are encouraged also, especially the members of the review panel, many of whom are here, not all, but many. And we are, here, we also have some board members. And I want to say a special hello and thank you for joining us to our founder, Sylvia Capolino. Sylvia, if you wouldn't mind waving, I usually don't deviate from the script, but I have to say thank you to Sylvia for having the biggest vision of any small person I've ever known. She's not a huge woman, but she's got great ideas. And so I want to wrap this up. And just on behalf of the Breast Cancer Coalition and our board of directors, I want to congratulate both of our award recipients, Dr. Sagar R. Shah and Dao Dai. And we wish you much success with these projects. We look forward to hearing about your progress. As Helene indicated earlier, we hope to visit you either virtually or in person, come to the lab, or at least get a camera in there to see how you're you're doing and learn more about the project. And I want to thank the review panel. I think this is another unusual aspect of this review panel in our breast cancer research initiative is that when funds get donated to this initiative, every penny goes to the researchers. We don't take out fees for administrative overhead, and we limit greatly what the institution is allowed to take from these awards as well. We really want to see funds raised go to the researchers who are working so hard, and especially after things like global pandemics, the funding becomes even more restricted than usual. But thanks to the review panel for this round of grant awards, the work that they put into this one. And I also want to express appreciation to the members of the entire research committee for their work and support throughout the year. Only a handful actually participate in review day, but the others are there right with us, working day in, day out, all year long. And if the review, if the research initiative or the research committee would just give a wave, people could see that you are here in great number today. And I also want to thank the coalition staff members who give so much of their time to furthering the coalition's mission. And I especially want to thank Michelle Lindsay, who you can't see. She's the woman behind the curtain. Thank you to Michelle, our office manager, for all you do to ensure the technological success in this and all of our events. So thanks to everyone for coming today. We hope that next year we'll be able to meet in person, but this year, if it's taught us anything, it's that virtual is the way to go in a lot of cases, and it's definitely expanded the ability for some of our friends to attend. I see friends from Buffalo and Ithaca, Bob Ritter, who is a great friend and colleague, so many of you. It's just wonderful to see you. Oh, Dr. Das is here. He has actually received an award from the Breast Cancer Coalition in the past. And it's just so nice that we could all be together today, even virtually. So I understand it's a beautiful day outside. It's hard to believe that that it's April and pretty nice out. So I encourage you all to get outside and do a nature immersion somewhere. Thank you. You're clapping somewhere in the building. I don't know if you can hear that, Helene, but that's great.
Okay. Well, again, Sagar, Tao, thank you so much for taking the time. This was, I don't know if you saw that in the chat, there were several people who commented great presentations and thanked you. So you really were, you were right on the money with presenting to a primarily lay audience, and we really appreciate that. So I will continue to be in touch. And
thank you very much for the opportunity, and again, deeply honored, and looking forward to working with the coalition, and at some point also learning more about the work that Tao and his group also pursue. But thank you again.
Thank you.
Enjoy your day.
And yeah.
It's really an honor to receive the award. Yeah. Thank you.
Okay.
Thank you.
Thank you.
Okay, Tom. Okay.